Hello and welcome, dear students. Today we are going to discuss about the topic transcription. The main objectives of today's lecture are to know about the basic concept of transcription, to understand the functions of transcription, to understand the underlying mechanism of process of transcription, and finally to know about the post-transcriptional modifications for production of mature mRNA. Dear students, let's start with the introduction first. As we all know that the discovery of DNA was one of the most significant findings in the field of science. Today, deeper insight into the DNA structure has unraveled answers to questions that have been around for a long period. The central dogma explains how the DNA codes for the proteins which proceed in three stages, namely the replication, transcription, and translation. Once DNA replicates into two strands, the information is copied into RNA by the process called transcription. Let's learn more about the process of transcription in detail. First, we will have an overview of transcription. Transcription is the first step in gene expression in which information from one gene is used to construct a functional product such as a protein, which may be a structural one or an enzyme. The goal of transcription is to make an RNA copy of a gene's DNA sequence. For a protein coding gene, the RNA copy or transcript carries the information needed to build a polypeptide protein or a protein subunit. Eukaryotic transcripts need to go through some processing steps before translation into proteins. The main enzyme involved in transcription is RNA polymerase, which uses a single-stranded DNA template to synthesize a complementary strand of RNA. Specifically, RNA polymerase builds an RNA strand in the 5' to 3' direction, adding each new nucleotide to the 3' end of the strand. Dear students, let's now have an introduction on to the genetic information. Genetic information flows from DNA into protein, the substance that gives an organism its form. This flow of information occurs through the sequential process of transcription, that is DNA to RNA conversion, and translation, that's RNA to protein conversion. Transcription occurs when there is a need for a particular gene product at a specific time or in a specific tissue. During transcription, only one strand of DNA is usually copied. This is the template strand, and the RNA molecules produced are single-stranded messenger RNAs, that's mRNAs. The DNA strand that would correspond to the mRNA is called the coding or sense strand. In eukaryotes, that is the organisms that possess a nucleus, the initial product of transcription is called a pre-mRNA. Pre-mRNA is extensively edited through splicing before the mature mRNA is produced and ready for translation by the ribosome, the cellular organelle that serves as the site of protein synthesis. Transcription of any one gene takes place at the chromosomal location of that gene, which is a relatively short segment of the chromosome. The active transcription of a gene depends on the need for the activity of that particular gene in specific cell or tissue, or at a given time, small segments of DNA are transcribed into mRNA by the enzyme RNA polymerase, which achieves this copying in a strictly controlled process. The first step is to recognize a specific sequence on DNA called a promoter that signifies the start of the gene. The two strands of DNA become separated At this point, an RNA polymerase begins copying from a specific point on one strand of the DNA using a special type of sugar-containing nucleoside called ribonucleoside 5 triphosphate to begin the growing chain. Additional ribonucleotide triphosphates are used as the substrate, and by cleavage of their high-energy phosphate bond, ribonucleoside monophosphates are incorporated into the growing mRNA chain. Each successive ribonucleotide is directed by the complementary base pairing rules of DNA. For example, a cytosine or C in DNA directs the incorporation of a G, that is guanine, into the RNA. Likewise, a guanine in the DNA is copied into a cytosine in RNA. That's a thymine into adenine and adenine into a uracil. RNA contains uracil in place of the thymine of DNA. Synthesis continues till a termination signal is reached at the end point of the RNA polymerase drops off the DNA and the RNA molecule is released. Ahead of many genes in prokaryotes, organisms that lack a nucleus, there are signals called operators where specialized proteins called repressors bind to the DNA just upstream of the start point of transcription and prevent access to the DNA by RNA polymerase enzyme. These repressor proteins thus prevent transcription of the gene by physically blocking the action of the RNA polymerase. Typically, repressors are released from their blocking action when they receive signals from other molecules in the cell, indicating that the gene needs to be expressed. Ahead of some prokaryotic genes are signals to which activator proteins bind to stimulate transcription. Dear students, let's now discuss the functions of transcription. Life on Earth is said to have begun from self-replicating RNA, since it is the only class of molecules capable of both catalysis and carrying genetic information. With evolution, proteins took over catalysis because they are capable of a greater variety of sequences and structures. Additionally, the bonds on the sugar phosphate backbone of RNA are vulnerable to even mild change in pH and can undergo alkaline hydrolysis. Therefore, DNA emerged as the preferred molecule for carrying genetic information since it is more stable and resistant to degradation. Transcription maintains the link between these two molecules and allows cells to use a stable nucleic acid as the genetic material while retaining most of their protein synthesis machinery. In addition, separating DNA from the site for protein synthesis also protects genetic material from the biochemical and biophysical stresses of complex multi-layered processes. Small errors in the RNA transcript can be overcome since the RNA molecule has a short half-life, but change to the DNA becomes heritable mutations. In addition, transcription adds another layer for intricate gene regulation, especially in species with large genomes that require minute adjustments in metabolism. In eukaryotes, transcription also plays an important role in transferring the information from DNA to the cytoplasm because the nuclear pore is too small to allow ribosomes, proteins, or chromosomes to pass through it. While the nuclear pore has a diameter of about 5 to 10 nanometers, ribosomes are between 25 to 30 nanometers in size. Many proteins are wider than 10 nanometers, and fully condensed chromosomes can be over 2000 nanometers in size. Therefore, primary machinery for protein synthesis cannot enter the nucleus and stretch of DNA cannot exit the nucleus. Dear students, let's now talk about the mechanism of transcription. Transcription creates a single-stranded RNA molecule from double-stranded DNA molecule. Therefore, only the information in one of the strands is transferred into the nucleotide sequence of RNA. One strand of the DNA is called the coding strand and the other is the template strand. Transcription machinery interacts with the template strand to produce an mRNA whose sequence resembles the coding strand. Other names for the template strand include antisense strand and master strand. Two different genes on the same DNA molecule can have coding sequence on different strands. Transcriptional activity is particularly high in the G1 and G2 phases of the cell cycle when the cell is either recovering from mitosis or preparing for the dramatic events of the next cell cycle of cell division. The process of transcription begins when an enzyme called RNA polymerase or RNA pol attaches to the template DNA strand and begins catalyzing the production of complementary RNA. Polymerases are large enzymes composed of approximately a dozen subunits, and when active on DNA, they are also typically complexed with other factors. In many cases, these factors signal which gene is to be transcribed. Three different types of RNA polymerase exist in eukaryotic cells, whereas bacteria have only one RNA polymerase. In eukaryotes, RNA polymerase 1 transcribes the genes that encode most of the ribosomal RNA, that is the rRNA. And RNA polymerase 3 transcribes the genes for one small rRNA, plus the transfer RNAs that play a key role in the translation process as well as other small regulatory RNA molecules. Thus, it is RNA polymerase 2 that transcribes the messenger RNAs which serve as the templates for the production of protein molecules, that is the translation. Dear students, let's now talk about the transcription initiation. The first step in transcription is initiation when the RNA polymerase binds to the DNA upstream, that is 5' of the gene, at a specialized sequence called a promoter. In bacteria, promoters are usually composed of 3 sequence elements, whereas in eukaryotes there are as many as 7 elements. In prokaryotes, most genes have a sequence called the Pribno box, with the consensus sequence, that is TA, TAAT, positioned about 10 base pairs away from the site that serves as the location of transcription initiation. Not all Pribno boxes have this exact nucleotide sequence. These nucleotides are simply the most common ones found at each site. Although substitutions do occur, each box nonetheless resembles this consensus fairly closely. Many genes also have the consensus sequence TTGCCA at a position 35 bases upstream of the start site. And some have what is called an upstream element, which is an AT-rich region 40 to 60 nucleotides upstream that enhances the rate of transcription. In any case, upon binding, the RNA polymerase, that is the core enzyme, binds to the another subunit called the sigma subunit to form a hollow enzyme capable of unwinding the DNA double-stranded helix in order to facilitate access to the gene. The sigma subunit conveys promoter specificity to RNA polymerase enzyme. That is, it is responsible for telling RNA polymerase where to bind exactly. There are a number of different sigma subunits that bind to different promoters and therefore assist in turning genes on and off as conditions change. Dear students, eukaryotic promoters are more complex than their prokaryotic counterparts, in part because eukaryotes have the aforementioned three classes of RNA polymerases that transcribe different sets of genes. Many eukaryotic genes also possess enhancer sequences which can be found at considerable distances from the genes they affect. Enhancer sequences control gene activation by binding with activator proteins and altering the 3D structure of the DNA to help attract RNA polymerase second enzyme, thus regulating transcription. Because eukaryotic DNA is tightly packaged as chromatin, transcription also requires a number of specialized proteins that help make the template strain accessible. In eukaryotes, the core promoter for a gene transcribed by RNA polymerase II is most often found immediately upstream of the 5' start site of the gene. Most polymerase second genes have a Tata box, that is consensus sequence of TA, TT, AA 25 to 30 bases upstream of the initiation site, which affects the transcription rate and determines location of the start site. Eukaryotic RNA polymerases use a number of essential cofactors collectively called general transcription factors, and one of these, that is TF2D, recognizes the Tata box and ensures that the correct start site is used. Another cofactor, that is the transcription factor 2B, recognizes a different common consensus sequence GC, 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 CC approximately 38 to 32 bases upstream. In eukaryotes, genes transcribed into RNA transcripts by the enzyme RNA polymerase II are controlled by a core promoter. A core promoter consists of a transcription start site, a Tata box at the 25 region, and a transcription factor 2B recognition element at the 35 region. The terms strong and weak are often used to describe promoters and enhancers according to their effect on transcription rate and thereby on gene expression. Alteration of promoter strength can have deleterious effects upon a cell, often resulting in disease. For example, some tumor-promoting viruses transform healthy cells by inserting strong promoters in the vicinity of growth-stimulating genes, while translocations in some cancer cells place genes that should be turned off in the proximity of strong promoters or enhancers. Enhancer sequences do what their name suggests, that is, they act to enhance the rate at which genes are transcribed, and their effect can be quite powerful. Enhancers can be thousands of nucleotides away from the promoters with which they interact, but they are brought into proximity by the looping of the DNA. This looping is the result of interactions between the proteins bound to the enhancer and those bound to the promoter. These proteins that facilitate this looping are called activators, while those that inhibit it are called repressors. Transcription of eukaryotic genes by polymerase I and III is initiated in a similar manner, but the promoter sequences and transcriptional activator proteins vary. Dear students, let's now discuss about the transcription elongation. Once a short RNA oligonucleotide of more than 15 bases is formed, RNA polymerase proceeds along the template DNA strand. The transcript is identical to the coding strand except that the nucleotide backbone has ribose sugar instead of the deoxyribose sugar of the DNA, and adenine base pairs with the uracil instead of the thymine in the DNA. RNA polymerase can catalyze the formation of a phosphodiester bond between the fifth carbon atom of an incoming nucleotide and the third carbon atom of the last nucleotide in the existing transcript. Since the RNA molecule has a free phosphate attached to the fifth carbon on the first nucleotide and a free hydroxyl group on the third carbon of the last nucleotide, RNA is said to be transcribed in a 5-3' direction. Dear students, let's now discuss about the transcription termination. Unlike DNA replication, where the DNA polymerase continues to add nucleotides till it reaches the end of the molecule, transcription has to be terminated at a particular location for effective gene regulation and expression. Terminator sequences are found close to the ends of non-coding sequences. Bacteria possess two types of these sequences. In RHO, that is row-independent terminators, inverted repeat sequences are transcribed that can then fold back on themselves in hairpin loops, causing RNA polymerase to pause and resulting in release of the RNA transcript. On the other hand, RHO or row-dependent terminators make use of a factor called row, which actively unwinds the DNA RNA hybrid formed during transcription, thereby releasing the newly synthesized RNA chain. In eukaryotes, termination of transcription occurs by different processes depending upon the exact polymerase utilized. For polymerase 1 genes, transcription is stopped using a termination factor through a mechanism similar to row-dependent termination in bacteria. Transcription of polymerase 3 genes ends after transcribing a termination sequence that includes a polyuracil stretch by a mechanism resembling row-independent prokaryotic termination. Termination of polymerase 2 transcripts, however, is more complex. Transcription of polymerase 2 genes can continue for hundreds or even thousands of nucleotides beyond the end of the non-coding sequence. The RNA strand is then cleaved by a complex that appears to associate with the polymerase. Cleavage seems to be coupled with termination of transcription and occurs at a consensus sequence. Mature polymerase 2 mRNAs are polyadenylated at the 3' end, resulting in a poly A tail. This process follows cleavage and is also coordinated with termination. Dear students, let's now discuss the post-transcriptional modification for production of mature mRNA. The important post-transcriptional modifications that take place for the production of mature mRNA are: the first is capping at the 5' end. The capping of pre-mRNA is carried out by the addition of 7-methylguanosine to the 5' end. This is achieved by the removal of terminal 5' phosphate that is done with the help of phosphate enzymes. This reaction is accelerated by the enzyme adenosyl transferase, which at last produces diphosphate 5' end. The 5' end product thereafter attacks the alpha phosphorus atom of the GTP molecule. This enzyme called guanine L helps to transfer a methyl group from adenosyl methionine to the guanine ring. This type of ring is called a cap-0 structure. Some of the functions of the 5' cap are as follows: enhanced translation, protection from some ribonucleases. It also enhances the transport from the nucleus. It also helps in enhanced splicing of the first intron for some pre-mRNAs. Dear students, let's now talk about the maturation at the 3' end, that is cleavage and polyadenylation. When a string of adenine nucleotides is added with a 3' end of the primary transcript, it's known as polyadenylation. Proteins are the ingredients required for polyadenylation, new script, and cleavage. Proteins required for cleavage of pre-mRNA are cleavage and polyadenylation specificity factor, that is CPSF. Cleavage stimulation factor, that is CSTF. Cleavage factors 1 and 2, these are RNA binding proteins. Polymerase A and RNAP second. The termination of transcription cannot be carried out by RNA polymerase second at the fixed position. However, they often transcribe beyond the 3' end of the mature mRNA. The correct 3' end is generated by specific endonuclease cleavage, addition of 250 adenosine in the form of a poly A tail is done. This tail helps to confer resistance to 30 exonucleases and also helps in the efficient translation initiation. The functions of the poly A tail are that it promotes mRNA stability, it promotes recruitment by ribosomes, and it also helps in synergistic stimulation with capping and also enhances translation. Dear students, the sequence of 3' polyadenylation. A cleavage sequence CA, polyadenylation signal sequence AA, UAAA, GU, rich sequence present in the 20-40 nucleotides, which is downstream to the cleavage site. Now let's discuss about the splicing mechanism. Introns are the most intervening sequences which interrupt the sequences of mature mRNA, and these are usually non-coding. However, on the other hand, introns like 1 to 19 have the characteristic to do code. Introns are highly reactive and get spliced upon the RNA, assuming the secondary structure after transcription process. These sequences are untranslatable because they remain absent in the mature transcript. For better understanding, splicing is a process of joining the exons by a pre-mRNA sequence. As a result, the introns are eliminated. Mostly, the introns are present in eukaryotes and rarely found in prokaryotes. Introns show varied variations in their length and number with an average size of 3.3 kb in human beings. In addition to generating functional mRNAs, splicing assists in the export of mRNAs to the cytoplasm. Dear students, let's now discuss the mechanism of splicing in detail. Splicing of introns is carried out in the nucleus with the help of a precise and complex arrangement of protein and ribonuclear substances. With the assistance of splicing mature mRNAs to the cytoplasm, this process involves around 70 proteins in higher eukaryotes and 5 fewer ribonucleic protein particles. Thereafter, pre-RNAs, that is exons and introns, are processed to form an mRNA containing only exons while the introns have been removed. In this mechanism, two different types of splicing takes place as self-splicing and spliceosome-mediated splicing. Dear students, let's now discuss about RNA polyadenylation. Post-transcriptional modification of RNA plays a great role in the various biological processes. Polyadenylation is considered a well-conserved regulatory mechanism in the involvement of eukaryotes and endonucleolytic cleavage along with adenosine monophosphate units on 3' ends, resulting in the formation of a poly A tail. Dear students, this was all about today's lecture of transcription. See you next time with a different topic. Till then, take care. Bye bye.